Hello, welcome to my poster. The poster is titled A Unitig-Centered Pan-Genome Approach for Predicting Antibiotic Resistance and Discovering Novel Resistant Genes in Bacterial Strains. We are from the Graduate Institute of Biomedical Informatics at Taipei Medical University. So about the background, so far the antimicrobial resistance is gradually becoming one of the medical problems, especially in the infectious diseases. And the collection of non-antimicrobial resistant genes, according to our past studies, is still not comprehensive enough for building a very accurate prediction model. So in this study, we propose a Unitig-based pan-genome approach for building the antimicrobial resistance prediction model and for analyzing the potential antimicrobial resistance genes. Here is the workflow. So our genomes are collected from the public repository. We collected the genomes from the Patrick database, and so we obtain the microbial genomes, and from the genomes we firstly did a quality check, and then we built a compacted deburring graph in order to extract the Unitig sequences from the compacted deburring graph. And after that, we built the presence and absence table of the Unitig across the collected genomes or the strands. So here you can see that here these are the strands and here are the Unitig, and then each Unitig are marked as 1 or 0 in the presence and absence table. And we not only build the table for the training dataset, which is from the Patrick dataset, we also obtained a external validation dataset from another published study. And the training dataset consists of 1,106 strands, and the validation study consists of 414 strands. And after that, we conducted a feature filtration and feature selection in order to select the most meaningful features for building the prediction model. And then we validated our model through both conducted the tenfold cross-validation on the training dataset as well as from the external validation dataset that is not involved in the feature selection and model building process. And finally, we checked the genes exist on the selected Unitigs and see what their functions are. So the first thing that we observe is that the Unitig pan-genome is a closed pan-genome. As shown from this reflection curve, we have almost reached a saturation of the Unitig, suggesting that we have obtained most of the genome sequences for the this bacterial species. And we also conducted a antimicrobial resistance prediction performance evaluation, and we see that since our feature selection was conducted by XGBoost, we see that the XGBoost selected Unitig performs much better than the all-contact dataset and the Unitig with the non-antimicrobial resistant genes. And this phenomena exists for all 8 tested antibiotic resistance, suggesting that the XGBoost is capable of selecting the features that can be used for building a much more accurate model for antimicrobial resistances. We also inspected the genes that exist on the selected Unitig, and what we see that there are quite a number of genes that appear in more than 2 drug resistances, as shown in this plot. Here are the genes that exist in only 1 antibiotic resistances, and here are genes that exist in 2 antibiotic resistance. Here are 3 and 4, 5, 6, 7, and there are 9 genes exist in all 8 tested antibiotic resistances, and these are the names of the genes that exist in all 8 resistances, including one hepatotic protein, the exoprotein, and DNA gyrase, VGRG, and the integrase, so on and so forth. Most of the genes are not currently known to be contributing to antibiotic resistance, but because they are selected by all 8 antibiotic resistant data set, they're probably doing something in the antibiotic drug resistance mechanisms or regulations. Here is another plot that shows the co-resistance gene distribution, and you can see that there are this is a large exo protein that was also shown in the previous plot, and there are probably 30 or 40 of them that exist in more than 2 drug resistance data set. So we believe that these genes may warrant further investigation to see whether they really play a role in the antibiotic resistances. So here is the reference. The paper was published in 2024 on the Computational and Structural Biotechnology Journal.